when my mother heard that I'm colleague with Stephen Hawking, she was very impressed, though I've never seen him here in person. So I'm Cristian Picioreanu. I'm coming from Delft in the Netherlands. These are some pictures from nowadays Delft and 350 years ago Delft, painted by Jan Vermeer. And I'm a chemical engineer working with computational methods, applying numerical methods to biological and biotechnological systems. And actually this makes things a bit borderline, because the work is not mathematical enough to be appealing to mathematicians, I guess, or maybe not rigorous enough. It may not be scientific enough for biologists, or it may be too numerical for biologists. And on the other hand, it's not practical or applied enough for engineers. So I'm somewhere in the middle, which brings also advantages, because you can communicate with all these groups, hopefully. And we study mainly the biofilm development with models, different systems, biofilm ecology, as you can see here, and bacterial motility. We had some projects, also clogging of membrane applications, also porous media. I will talk about this during the third workshop, I think it's called, here. Dental biofilms, maybe somewhere in November I can tell you more about this topic, which is quite interesting. Biofilm fluid interaction, and today I will try to introduce you some work on electroactive biofilms. Some people call it in different ways, electrogenic, electroactive, and so on, different names for the same things. Practically biofilms that conduct current and transfer electrons to or from an electrode. And this has a few applications. Biocorrosion, or microbially induced corrosion, is what we would call the beast. We don't want to have these. Biofilms developing on oil pipes can damage, of course, the pipes, induce pitting corrosion. Then the good things about interactions between biofilms and substratum, electrochemical interactions, microbial fuel cells, which can produce some amount of current with biofilms, and newer, these current-producing sediments discovered in Denmark. And I will try to introduce you all these three topics and models we are working on for these three. First of all, I think I want to tell you about the differences between these systems. So they all contain some biofilms, so aggregates of cells living attached to some surface. In biocorrosion, you typically have iron dissolution, as ferrous iron here, below the microbial colonies. And then you need also to have cathodic places where oxygen is reduced. So iron dissolution is the anodic process. All these electrons must go somewhere, and they go on the same piece of metal to places where oxygen is available. So practically in biocorrosion, you have on one piece of metal, on one support material, conductive, you have both the anodic places and cathodic places simultaneously. Only that some anodic places are more differentiated, and in other zones you have more cathodic-like places. In microbial fuel cells, people separate these two places, the anode from cathode. You have an anode here, an electrode, where bacteria donate electrons, and you have a cathode, where electrons are accepted, come from this cathode and will produce either oxygen reduction or hydrogen evolution in bioelectrocatalysis, actually. So they are separated. And the electrons flow, of course, through the wire, so to say, flow. And at the same time, you have protons which have to go through some bridge or membrane from anode to cathode. So the electrodes are now separated, unlike in biocorrosion where they are on the same surface. Typically in biocorrosion, we want to predict corrosion rates, while in microbial fuel cells, we want to predict electrical currents obtained from substrates using these bacteria. And recently it was discovered that we can have also conductive biomass or biofilms, like, for example, these filamentous sulfide oxidizing bacteria, the sulfobulba, discovered somewhere in Denmark, I think is Nielsen group, Aarhus, and also direct interspecies electron transfer. So practically you have a conductive structure here, and some cells function as anodes, others as cathodes on the same piece of wire. And I will try to introduce you this later. What we want to predict here usually growth rates for these cells. How do they benefit from this community? Some cells give electrons to the conductive structure, other cells accept the electrons in cathodic places. So about corrosion, so I will have three topics here, A, B, and C. Now, what is biocorrosion? There are a lot of myths about biocorrosion, and everything that corrosion people cannot explain normally just with chemistry, they call it biological corrosion, which is not really the case. So this is localized corrosion as a result of microbial activity, different types of activity. It can be sulfate-reducing bacteria, so in anaerobic environment. There could be acid-producing bacteria, like these sulfide oxidizers. Iron oxidizing bacteria, they eat iron, so to say, like the Gallionella. Or you can have differential aeration cells. And these are in aerobic environments. So practically you can have microbial corrosion or microbiologically influenced corrosion, both in anaerobic and aerobic environments. And I will try to show you a little model on differential aeration cells. Now imagine this is a metal, iron, steel. It has a passive layer here. There is a colony of aerobic bacteria growing on this piece of steel, consumes substrates, consumes oxygen, forms biomass. You write some bacterial growth kinetics here, reaction, stoichiometry. Then you have electrochemical processes occurring on the surface here. You should have some anodic process, which is the metal oxidation and release of ions in the solution. You should have some cathodic space on this metal where oxygen is reduced to hydroxyl. So these electrons now go to the metal and will provide here oxygen reduction. On the other hand, you have some bulk reactions like ferrous ion oxidation, quite quick in water, and then precipitation because you may create an alkaline environment in this case. So you have rust as well. Now, if you try to put all these reactions in a closed system and also simulate the transport of ions and the transport of electrons through the metal, what you get typically with these simulations is the current distribution on the metal surface. So imagine this white patch here. This is the biofilm, a few colonies of bacteria, and this is the metal surface. So if we remove the biofilm, we can see that under this colony you have anodic currents. Anodic currents, these with red and arrows pointing up, means that you release ferrous ion in the solution under the colony. And you should have also enough cathodic space so that oxygen is reduced. And these are the arrows pointing down here. And what is important in biocorrosion is that these currents must be balanced. You cannot lose electrons on the same piece of metal. So if you integrate these currents over the whole metal surface, it should give you zero in the end. The positive currents and the negative currents added over the whole surface should be zero. This is not a battery, so that you produce a net current here. And here I have a little animation. This is typical for my presentations. A little animation with what happens in time. This is the corrosion rate you will see here. White means less and black means more corrosion. Here you will see biofilm growth starting with 5 cells. They will make 5 colonies here. And this is the current density, practically is the corrosion rate integrated over the surface. So if you run this a bit, you will see how the cells grow, how you have a maximum in corrosion, and then how the corrosion current actually decreases after a while. So this is quite interesting. If we stop somewhere here, you will see large differences. Under the biofilm, you have a high current, high corrosion. This is pitting here, pitting corrosion. While outside, there is less corrosion. And this is practically the total corrosion, and all these error bars show you how non-uniform the corrosion is. So this is now pitting corrosion, localized corrosion. If the biofilm grows further, something very interesting happens actually, because the corrosion rate decreases now. And this is practically a project we had with the TNO. The Dutch people know this organization. They wanted to apply bacterial coatings to prevent corrosion, to mitigate corrosion. And there are people working in the world on these kind of topics. So practically, if you have a compact biofilm, you can have corrosion inhibition because you run out of cathodic space in this case. If you have patchy biofilm, so colonies here and there, you may have corrosion enhancement or pitting because you can balance enough anodic places for corrosion with enough cathodic space where oxygen is reduced. So this was kind of a nice finding in this piece of work, that practically biofilms, aerobic biofilms like these, can act in both ways, enhance or inhibit corrosion.
Here, do they have unlimited supply of carbon substrate?
Yes. Only oxygen was limiting here. Carbon was not a problem. Of course, this won't work in reality because you have contamination. This is kind of a pure culture of aerobic organisms. But in the end, you have the anaerobes colonizing exactly these gaps. And they can even enhance corrosion even more, sulfate reducers or so. So it doesn't really work in reality, but we had a project, as you said, we had the money. And I think it was interesting to study this relationship. There are even people who try to kill the unwanted bacteria with antibiotics. I think it's really not practical at all. I mean, you want to make this microbial coating self-healing. That was one of the things. If you scratch this microbial coating, it will block again. It's not like your car. If you scratch it with something with your keys, then it will remain scratched. But if you have a microbial coating, it will close again. They will grow. I don't think it's really practical because it will grow, but it will also entrap anaerobes. And then what do you do with this? But at least we could shed some light on the mechanisms. Okay, now about microbial fuel cells. This will be the largest part of my talk, actually. So you have a biofilm growing on an anode. It can be graphite, it can be platinum, it can be steel. And this biofilm, for example, converts a substrate into product. Let's say the substrate is acetate, to keep things simple, which means acetate is converted into carbon dioxide, and you produce from 1 mole of acetate 8 electrons going through the wire and 8 protons. And the protons should go to the cathode. And this is now the problem. If you want to produce high currents, which means a lot of electrons, you have also to produce a lot of protons. And all this compartment will be acidified. This is one of the biggest barriers for this kind of technologies. So you have different transport mechanisms. Substrate must be in the bulk liquid, transported to the biofilm, and then diffusion through the biofilm. conversion in the cells, and then electrons are released here. Then you have transport of protons through a membrane to the cathode, and then at the cathode typically you put oxygen, and this has also to be transported. There is a reaction rate here. So you see here at least 10 limiting steps. And this is the problem, actually, that you have many steps, and which one is actually limiting in your system. That's why a model would be useful, at least for engineers, to calculate, well, which is my limitation in this process. And again, depending on money, people started with anode, and they said, okay, we've done everything, now the limiting step is the cathode. And then the source of funding changes, and then people study cathode. Now, once they finish with the cathode, well, what about this membrane? This is the limiting step, others claim. So it's very much driven about money, but in my case, it's just because I like it. So models could detect possible bottlenecks in this system. That's why we make models, engineers at least. You should understand how this electron transfer actually works. There are different types of electron transfer. You can have soluble substances, these redox mediators, which take electrons from the cell, bring them to the anode, and deliver the electrons to the anode. Redox mediators like quinones, phenazines, even humic acids, and so on. So this is the flow of electrons from a reduced substrate like acetate there to the electrode. It can be endogenous, generated by the cells themselves, like Shewanella is able to do, or they can be supplied externally. So people throw there some mediator just to enhance this electron transfer. However, if you work with a continuous system, that's a waste, because this will leak out of the system. So you should work in a batch system or repeated batch in this way. You can revert the flow of electrons, and now imagine you have a biocathode, and now the electrons have to go like this. While the mechanisms are, well, being quite well understood now for the anode, how electrons are transferred from the cathode to the cell, this is not known. Bruce Logan works in this in America very much, Derek Lovely. They have different hypotheses. At the moment, people don't know exactly how this works for the cathode. And you can have direct electron transfer. Cells attached to this electrode can transfer direct electrons via some kind of cytochromes. You can have a longer range conduction. Again, theories here are competing. Derek Lovely thinks one of these, Yuri Gorby another one. So different people, different theories, whether they are nanowires or super exchange or copper conduction, electron hopping, whatever. We don't mind very much right now in the model. We just put conduction, Ohm's law, and we'll see what happens. And it can be conduction through metal oxides, which, for example, in sediments, they can exist, via this pili, or it can be a conductive polymeric matrix. So we started with the simplest system, actually, when we started these projects, experiments done some 30 years ago here in England. Very nice experiments, I must say. It's a pleasure to read these old papers. People didn't rush to publish. They did thorough experiments. They reported very well all the conditions. No supplementary material that it's always obscure in nowadays journals. You know, you have to condense everything in 4 or 5 pages and then 30 pages of supplementary material. Everything was reported, so I thought very nice data to model, to test. So they add glucose to the anode compartment. You have cells. You have this mediator. It was thionine in their case. Cells will bring the glucose, we'll degrade it, we'll give electrons to the mediator, the thionine. This reacts at the anode and comes back to the cell and so on. It's a cycle. It's a mediator. And what they observed that during a couple of hours, this is a batch system. So they inject the glucose, they measure current over, let's say, 4 or 5 hours, and then the current drops. And then you can imagine why. The cells consumed all the glucose, no more electrons to be transferred to the anode. So this is what we wanted to model first. And the model is very simple. It contains two components, practically. You have a biochemical part and an electrochemical part. The biochemical reaction takes place in the cells. They degrade the substrate with mediator, oxidized form. So this is electron donor, electron acceptor. And then the mediator diffuses to the liquid, to the electrode. We have an electrochemical reaction here, comes back to the cells. So for this, you can describe the rate with monokinetics. We thought double limitation would be good enough here. So electron donor, electron acceptor. While the electrochemistry is described by a Butler-Volmer electrochemical rate. If you are familiar a bit with electrochemistry, you heard about this Butler-Volmer or Tafel kinetics. Practically, what this rate says is that the electron production here depends on the concentrations of mediators, also on the pH, because there is some hydrogen proton here. But the rate depends also on the overpotential. So this is the difference between the electrode solution potential and the redox potential of the mediator standard. So I will explain you a bit later about this. So we had to calculate this activation over potential, which is a function also, interestingly, of the current intensity. So if you want to calculate the current intensity, because this rate is linked with the current via the Faraday equation. So the current depends on the current itself. So it was kind of an implicit equation here. But it can be integrated within the solver. And then you put substrate, you put mediator in this solution, you have mass balances in a batch system, you solve this with these kinetics and see what happens. This is what happens. The substrate is the red curve here. Substrate is degraded in a matter of hours, 2, 3, 4, 5 hours. And then this is the amount of oxidized mediator that you produce. From this, you can extract the current. How many electrons this glucose delivered to the electrodes, to the anode. And this is the current together with the measured data. Of course, we had to fit some parameters. For example, the affinity constants. No one knows how much is this. But it looked quite okay in the sense that you have this plateau, quasi-plateau, for a few hours, and then the current drops when the substrate is limiting. So this is a batch system. If you integrate the current in time, then you get the total charge. They measured also the total charge. And there were also repeated batch experiments that they reported, which I liked very much, because after a few hours, when the substrate ends, they inject again glucose. And then you have again current. The black one is the current. And then the current drops again as the substrate is consumed. After 18 hours or so, they inject again glucose, you get again current, cells consume the glucose, produce current, and then it drops again.
Where's the hydrogen going?
The hydrogen goes to the cathodic compartment via a membrane.
Okay.
It's an Affion membrane. What you can see here that the current actually doesn't go to zero. There is something here producing current, and we think it's endogenous metabolism, some maintenance thing that takes place in the cells, or it could be storage from the storage products. They eat the glucose quickly and they store it and then when there is no more substrate, they will slowly degrade their internal reserves. You can say storage compounds or endogenous metabolism. If you put some endogenous metabolism, then of course you can fit this quite nicely. So you have here a base level.
So do you get more and more biomass in these systems?
This one didn't grow in this time frame of 24 hours. In this case, no. But in biofilms, yes, you get more and more biomass. This is what I'm going to show you.
But I mean,
but
can
not
they
here.
repeat? How often can they repeat this?
They did not put growth, how to say, other substrates. They didn't put, for example, a nitrogen source or so, in this case.
But if the cells don't grow?
They don't grow, no. But they metabolize. But, well, actually, I don't know if they divide or not, but from the curves here, it doesn't look like they grow, because the second peak here should be higher now, if there are more cells. The third peak now should be even higher. They lack some growth factor in this experiment, or they grow so slowly that you don't see it. Otherwise, the currents should be higher and higher all the time. And this is what you see actually in biofilms. That's what I have here, biofilms. Same mechanisms, you need the same biochemical model here in bacteria. Now the mediator will be oxidized or reoxidized at the electrodes, so you need the same electrochemical model. You need some model to grow the biofilm. This is how I explained you last time, during that COMSOL course. Solute transport in the biofilm, diffusion, reaction, electromigration probably. You need to know something about the mixing of this bulk liquid, the transport of protons or other ions through the membrane, also important, and of course some electrical model for your circuits. So this is what I like about these systems. It combines biology with chemistry, with physics, electricity, electrochemistry. It's quite, interdisciplinary. And actually people study this from some point of view. It's either the biochemistry or it's only the electrical component. Other people study only the membrane. A model can bring all this together. Whether this will be useful or not in the end, don't know.
Just so that I don't get too lost. So what do you actually build a membrane like this out of so that it doesn't biofoul?
It will foul.
Okay.
It will foul. There are cells also growing attached to the membrane.
Okay, so they eventually just, the membrane
Yes,
down.
yes, it does. There are now systems where they try to eliminate the membrane at all and make this cathode in contact with air directly. So there are different types of systems right now. But yes, cells will also foul the membrane. Okay, this I showed you before, actually. So for the biofilm growth, you can have continuum-based models. Advantage is, of course, that you can solve only differential equations based on transport and reaction. You can have also individual-based models like this. So you can represent now individuals, and disadvantages, they are still empirical in the sense that the transport of cells is not described by these continuous equations like convection and diffusion. That's why I put here the black box. This is not a PowerPoint failure or graphical card. Don't blame Microsoft again. This is my black box because we don't care somehow how the transport is done as long as it looks realistic. Should I say this at Cambridge? It does work, and actually this was a phototrophic biofilm for Jeff. These cells were growing when there is light, so the green cells.
Oh, so that's why you have this dark.
Yes. So now it's night, only the blue and the red nitrifiers are growing, and during the day the phototrophs are growing. So this was during another European project. Nice. Okay, and you can combine now the continuum description for the polymers with individual cells in the biofilm. We did this once and, well, no one followed on this direction. Though I still think this is an interesting way to go. Okay, so if you put all this together, what happens with the biofilm? You will see in this graph the substrate and the current evolution in time, and here you will see the biofilm thickness. And as the substrate is consumed in a batch system, biofilm thickness increases. It will reach here some 25 microns from this batch in about, I don't know, 8 hours of substrate consumption, which is fine. I mean, it's normal. You add substrate, cells take it up, they grow. At the same time, you have current production, which will reach a peak somewhere here where there is a maximum substrate consumption rate. Where this slope is the steepest, you have the maximum current production, practically. And the explanation is that first you are biomass limited. So first, not enough cells attached to the electrode and didn't grow enough, so you have just little current here. So the current increases now in parallel with the biofilm thickness increase. Later, you reach substrate limitations. So if the biofilm grows thick enough and you don't have substrate anymore, the current drops like this. And this is practically what people observe in their experiments. Again, model and experimental data. So this is now another time frame of 1 week. Again, typical for people working with microbial fuel cells on a short time, 7-day experiment. This COD is the organic substrate, probably you know this. And these are some famous experiments performed about 10 years ago in Derek Lovely's group. They had this geobacter biofilm consuming acetate, and they added acetate when they observed there is no more current production. This time is with biofilm, and you see here how the current actually increases after the second addition. And then it drops because there is no more acetate, and it increases again, which means that these cells grow now. They form a biofilm, the geobacter, the red biofilm of geobacter, and the current increases after each addition. And this is what we simulate now. We take this data and we put the acetate in the 1 millimolar concentration. They add it here. The acetate is consumed. This is simulated now. And then you add again, and this time acetate is taken up quicker because there is a thicker layer of cells. You add another batch, and again, even quicker, you have the acetate uptake. And this is how the predicted current looks like from this acetate uptake. It doesn't fit the last part of the experiment. And we think this is because now the mediator actually, the diffusion of the biofilm is too thick somehow, so that you have too large diffusional limitation for the mediators. And I think this is quite true, is what Jan said, it's difficult to prove something negative or so. We wanted also to show that for geobacter, it doesn't work well with these soluble mediators. Geobacter actually can transfer directly the current with these wires, conductive biofilm. So if you have a conductive biofilm, you won't have this diffusion limitation in this case. So I wanted to show you here also the limitations again. Initially you have biomass limitation, small gradients, small current. The biofilm is the green part here, and this is the distance from the anode surface. So plenty of substrate and oxidized mediator, but the current is small because there is not enough biomass. Later in time, you have gradients of substrate, gradients of oxidized mediator. The current increases now. The biofilm is thick enough. It's a typical counter-diffusion system, like the one we studied in the COMSOL course. One part of the redox couple, the electron donor, comes from the liquid, diffuses in the biofilm, and the electron acceptor is generated here at the electrode. It's the oxidized mediator coming from the electrode. So it's counter diffusion in this case. So that's why you see this minimum here. Where they meet, you have the reaction taking place. And this is the reduced mediator exactly mirroring the oxidized form. And in the end, when the substrate is exhausted after 8 days of experiment, batch experiment, the current drops in this way. We studied also dual systems now, the competition with methanogens. So you have this electrogenic bacteria. We can calculate some approximate stoichiometry from thermodynamics, some yields practically, given a certain standard redox potential for this mediator. You know, you can translate everything in delta G's, and then you can estimate a bit the yields in this case. Now, if you have an efficient mediator, you have a lot of energy for growth and not too much electricity production, but the cells grow. You know, the potential for this mediator should be in between the two components of the redox couple, electron donor and electron acceptor. So depending on the position for the mediator potential, you can get little current but a lot of growth, or the other way. So this is an efficient.
No,
Yeah?
stop. Say that again. So, why
Yes?
should it be in the middle? I'm confused. So,
This mediator takes the electrons from the cells. So it should be higher than the substrate here. But it's less than the oxygen,
yeah,
because the oxygen is at the cathode side.
right.
You won't have driving force if this would be higher.
So that I get, but you actually mean, you just mean in between, or, yeah. So if you were to set up like a redox ladder, right, by having another species
Yes,
there,
yes, it would work. It could be transferred from species to species as well.
but thermodynamically, you'd like to make it as many steps as possible, right? Isn't that the logic?
It depends. This approach considered only one step,
Okay.
just to be clear. Yes,
Okay.
you would have a whole redox chain, of course, in the cells, Na, DH, cytochromes, all these things. But here is only one. Everything is lumped.
Okay.
Okay, here is the competition. So the electrogenic bacteria are black and the methanogens are red here.
Cristian, then
Yeah.
mediators, do they have to have electron potentials that are then more oxidized than
Then the substrate.
lowest? No, not
Then-
the substrate,
yes.
but then NAD plus.
Yes,
and
yes,
so on.
yes, also. So we just took here the electron potential for thionine, which seems to work with the cells used in these fuel cells, microbial fuel cells. Indeed, there is a range for this. Yeah. Okay, so you see the black ones, the electrogens dominate in this case. If you now decrease this potential, you have less energy for growth, but a larger part of the electrons will go to the wire. And in this case, the black ones, the electrogens, grow slower and the methanogens can populate the biofilm. This is what people don't want. This is always the problem with the anodic compartments of microbial fuel cells. How can you get rid of methanogens? How can you convince cells not to perform methanogenesis? Because we want to apply these systems in natural environments, wastewater treatment, to extract energy from wastewater. And then everyone is there. How can they outcompete the methanogens in this case? And now speaking about wastewater and complex environments, now this is of course some anaerobic digestion scheme ending with methane. And you have different communities here, the glucose consumer, the butyrate, propionate consumer, acetate, and then methanogens. And the point here is to hijack the electrons from acetate and give them to the electrode. Instead of producing methane, you want to produce electricity, and you have to couple this with some cathode. So you write, well, electrochemical reactions, of course, here, all kind of microbial reactions, like in anaerobic digestion, acid-base equilibria, because pH seems to be very important. Okay, I will jump a bit quicker over this boring part, probably. You make mass balances with Nernst-Planck equation, charge balance, a lot of compounds here. And what you get now is some biofilm with a certain microbial distribution. And these arrows are practically the fluxes of protons. So it's a batch system. Day 2, the biofilm is thin, and we put all these 5 types of microorganisms in equal amount initially, same ratio. And now you see how the glucose consumers, the red ones, dominate. They grow very fast, as long as they have glucose. After the glucose is exhausted, then you have the other steps in the anaerobic digestion. And then these are the acetogens, I think, the light blue, for example. So the proton flux here increases in time. You have acidification until the substrate is depleted here. So here it dies. So you can see also what happens. The biofilm grows, more and more protons associated with electron production on the base here. So this biofilm grows on some electrode. While protons are released in the medium, electrons go in the electrode. This is the whole point here. So you practically split the charge. Protons will go to the cathode and electrons through the wire. And you have very strong gradients here. That's quite interesting. Hydrogen is very important, H2. So you have these places, the red places where hydrogen is produced strongly, and next to them you have places where hydrogen is consumed. Blue means low concentration. So this community must be tied together somehow. This is a parenthesis, practically. We observed this also in other methanogenesis systems. You know these syntrophic acetogens, which grow with these hydrogenotrophs together. So again, we have these 5 types of microorganisms, from sugar consumers, propionate, butyrate, valerate, acetate consumers, and hydrogen consumer methanogens here. And we let them develop. Well, if you look here, let me stop this. So in the beginning, this was done for anaerobic granules for wastewater treatment. And we put everything in the same ratio here. So let's say 10 red cells, 10 green, 10 blue, 10 yellow, and so on. So the same amount. And now they grow. Glucose consumers grow very fast. But what you see here is very interesting. It's a nice result from individual base models. The blue and the green tend to stay together. There is no rule in the code, in the computer code, that tells these two to stay together. So you have to believe this. There is no special attachment or so. They are together because they exchange hydrogen. So practically the green one is the propionate consumer, which produces acetate. It gets thermodynamic inhibition. So they have to grow next to the blue ones, which consume the hydrogen. And I'm sure the microbiologists know these systems very well. I think when they were discovered, people didn't know there are 2 species. Much later they realized they are so tight together, and this is what the model can produce as well. So these clusters can be a result from interspecies transfer.
Why do you have this boundary that is still
This
red
one?
and then
This is decayed.
yeah, but then surrounding the others, you still have like a red shell or something.
This one, oh yeah, these are small cells that are not pushed fast enough. It's a model artifact.
Oh.
It's a model artifact. With this pushing balls model, this cannot be pushed too far away. So it's when these colonies grow, they will push small layers of cells, like like a bulldozer a bit. Okay, so going back to the current production, you see the typical phases for anaerobic digestion. This is glucose consumption. You produce here acetate, propionate, butyrate, but then acetate is consumed by the electrogens. And this coincides with the current production here. And pH is also interesting here. So first you have the anaerobic digestion, you form methane, you produce acid, the pH decreases, so it's acidified. And then you have the electrogenesis from acetate as a second step, and then you have a further dip in pH, because this also produces some protons. And the point is, how do you get rid of these protons to make the process feasible? Otherwise the cells will stop working. Well, one way is to buffer the system. These are carbonate buffers, different concentrations. The simulation even crashed at this point. That's why it ends up like this. It's too acidic, everything stops. If you put more CO2, it will stay more or less like this. But how can you add this in wastewater somehow? Carbonate, I don't know, limestone. And another mechanism would be the transfer through the membrane to the cathode. So if you don't have enough buffer, at least you should be able to transfer protons to the cathodic space. And this can also be very limited, because actually people noticed that it's not the proton which is transferred, but mainly the sodium ions. These membranes transfer any positive ions, any cations. And you know, proton pH concentration 10 to minus 7, sodium about 3 orders of magnitude more. So it's mainly the sodium transferred and not the proton. They are not very effective, these membranes. Okay, a last thing here.
Have you ever tried using just salt water versus fresh water? So you have a big...
I don't know if this is tried or not. People used to use wastewater.
They also tried sediments, no, in the ocean.
Yes, yes, sediments as well. I will come to the sediment in a minute. This is now the porous electrode. So there were experiments where I did this Marie Curie stage in Newcastle with flat electrodes and porous electrodes. You know, the concept is that the porous electrode should give more surface area per volume. And it should work better, but actually in reality it didn't work better. The flat electrode gave higher currents than the porous one. And the whole point stays here in mass transfer. So we did some simulations here. This is the acetate represented on this color scale, and these are graphite granules. Even if they are white here, you should imagine them black. That's what we do with computers. You can change colors immediately. So these are graphite granules, and these are colonies growing on the granules. At day 3, there are a few colonies now formed. There is no acetate limitation, but if you look after 5 days, these colonies blocked somehow the porous medium, and there is little acetate left here, deep in the biofilm. So this is the surface, and there is a flow. These white arrows mean flow. Flow calculated here, and there is transport mainly by diffusion within the porous medium. And black means that the cells are suffering. I tried to put some meaningful color codes, at least in the Western culture. So white are active cells and black are less active or inactive cells, because there is much less acetate here. So practically these porous electrodes are good only if you are able to provide the substrate deep in this porous medium. And well, here you see also the electron currents going to the graphite beads. And in this graph you can see the protons flowing out, and practically what you see here is an acidification inside the electrode. how do you get rid of this acid here now? This is a problem, because the cells here not only suffer because of lack of acetate, but also because of too low pH. So mass transfer is very important in this case, and many people don't realize this. It should be obvious, but it's not really. So probably with models we can convince them. Yeah, if you want to see some little animations here, you see here cells growing, colonizing the graphite beads. Red arrows are now electron currents, and the black arrows are protons. So what happens? These grains now, the graphite beads, get electrons, and the proton has to diffuse out of the porous medium and find its way to the cathode. And of course, these grains are connected. They all have the same potential here. Okay,
Why can it not be something like in the same structure that captures and electrons and protons or something?
In the same structure. I don't see how electrons can travel through water somehow.
No, I mean, if the electrons are captured, why do you have to send the protons all the way to another side and not
consume them
at the same side?
Yes, if you have buffers, of course they will be, or if there are other cells which can produce the cathodic process in place there. But somehow anode and cathode at the same place is not really nice.
I was thinking, I mean, maybe practically enormously difficult, but what if you would have two planar, an anode and cathode, and you have only a single cell layer?
Yes, people try this. So practically you want to say that the distance between cathode and anode should be reduced as much as possible.
Yeah, to a single cell layer.
Yes, people try this. On the other hand, if you have oxygen penetrating from the cathode, they won't do this. They won't transfer electrons to the electrode. They will take up the oxygen, no, as a terminal electron acceptor, not the electrode. So I think there are challenging problems here. And you know, a lot of money put in these microbial fuel cells, a big hype, but until now the practical applications are rather limited. And it will never compete with nuclear energy, by the way. Okay, last part. Probably you are interested in this as well. You can stay 5 more minutes. Sulfide oxidizing bacteria. You know the papers from Nielsen, Denmark, Aarhus. We have now kind of a model for their system. So imagine a sediment now. Oxygen coming from the top. This is the aerobic part, red. Sulfide coming from the bottom of this sediment. So you would have typically sulfide oxidizing bacteria somewhere in the middle where the sulfide meets the oxygen. So an oxygen gradient and a sulfide gradient, and the sulfide oxidizers should live happily at this place. You know, this is sulfide, oxygen, produce sulfate, produce protons, which means that you should have some acidification here. And that's why the pH should somehow drop a bit. And this is actually what people measure. Michael Kuhl in Denmark, for example, always uses this kind of electrodes, the oxygen electrode, the pH, the sulfide, to measure these gradients. So this is another typical counter-diffusional system. Oxygen from the bulk liquid, top of the sediment, sulfide coming from degradation from the bottom. And they meet here, and here is where sulfide oxidizers grow. Okay, that would be the typical example. And what people measured actually a few years ago now and published in 2 Nature articles, I think it's Nielsen, but I forgot which, all the Danish are Nielsen, one of them. They measured this kind of profiles, oxygen penetrating about 1 millimeter in the sediment depth, sulfide consumed here, and nothing in between, and also an alkalinity production here. You should have acidity, not alkalinity here. And we try to make a model for this just to see how this would work actually, to explain it. So this is the oxygen consumption place. This should be, well, I mean, not only this spot, but this area is the sulfide consumption. Here you have alkalinity production. Look at the pH here. It increases. It's more than 7 now. And acidity here is not measured practically. And they discovered these filamentous sulfide oxidizing bacteria. I think they belong to the desulfobulba, whatever, you know better. Desulfobulba. What is interesting, you have these cells in a filament and there is a conductive sheath. It's a tube. I imagine it like those light snakes you have for Christmas at the window. You have these light bulbs and then you have this plastic outside to keep the bulbs together, you know. So, but that's conductive now. And well, we try to make a model now so that we can explain these profiles and what is the effect of conductivity now and how long can these filaments actually grow until they are limited, either by substrates or by conductivity or other things. So we have the filament, substrate concentrations, and we try to put a split catabolism now. So half of the reaction is done by cells deep in the sediment, the sulfide oxidation to sulfate, and the cells here at the top perform oxygen reduction. So you can see here from the reaction, they consume protons here, so this should become alkaline, and they produce proton here, this should become acidic, this part. We split the catabolism, but there is one problem. How the electrons here have to be transferred somehow to this place. And the hypothesis is that this conductive sheath allows these filamentous bacteria to transfer electrons over a distance of centimeter or so, even. That's what they measure. And that's why we put an intermediate, again a mediator which exists in the cells, so it's not diffusible in the medium, but it's in the cells now. Electron is transferred to this conductive structure. Electrons will go to the cathode, taken here, and they perform oxygen reduction. So metabolism is now split in two parts. And we calculate now the delta G's for the two processes and see what happens in this case. So practically we have this anodic catabolism with some stoichiometric equation, electron donor, electron acceptor. The oxidized electron donor is the sulfate, the reduced electron acceptor is the reduced mediator. 8 electrons in this case, you know, from sulfide to sulfate, it's -2 up to +6. And some Michaelis-Menten kinetics, we thought it should be, well, a first approximation. The cathodic part, it's also represented by this. Electron transfer, so this step here. The mediator should accept electrons here being reduced, and here the mediator should transfer electrons to the wire and becomes oxidized. And this is represented again by a Butler-Volmer kinetics, reversible, with a certain potential of this mediator. And you can imagine there is now a potential difference between these two areas. Otherwise, it wouldn't work. And this potential difference arises from conductivity. So if conductivity is infinite, then it's the same potential everywhere, like in a metal. But this is, I think, the whole trick here. The conductivity is not infinite, so you have a potential favorable here for sulfide oxidation and here for oxygen reduction. And all the electrons are contained within the filament. You insulate the ends. So yeah, this is the local sheath potential, and this is the standard potential for, we thought, cytochrome C. We don't know what is actually transferring these electrons. We thought, okay, a reasonable approximation would be to take this cytochrome until people will tell us it's wrong. And we also add acid-base reactions because we need to calculate pHs as well. Okay, so the domain is one-dimensional. This is the filament, but the domain is longer because you need a diffusive boundary layer for sulfide and one for oxygen at the top. So you have solute transport, a few essential chemical species with ions because you need pH calculation, so you need to speciate a bit. There will be, of course, in the liquid phase, you have Nernst-Planck, so diffusion migration plus electroneutrality. You put a sulfide source at the bottom. So at this point in the domain, you put an oxygen source at the top. Then for the electron transport, we have Ohm's law in the conductive sheath. So the solute is transported everywhere in the domain. Cells or boundary layers, doesn't matter. While electron is transported only through the sheath here. So in the end, you get this kind of equations. Practically, it's well known. Current production and consumption. And you insulate the ends of this filament, which means zero flux here of electrons. And the filament grows now. It's an advection reaction equation. And it grows because we put inside also an autotrophic metabolism. So these catabolic reactions generate energy for the cell to grow. And we assumed that it grows on CO2. It's autotrophic. We don't know. This is an assumption here. And it seems to be a strong assumption, and it seems to be a wrong one, I think. But let's say they are autotrophic. So the energy they get from this redox couple, they will use to create new cells from CO2. So if you put this, you will get some velocity by which these two tips of the filament will advance, down, up. And then the mediator, this cytochrome, is also dividing in the cells. So it's, not divided, produced in the cells, and it will spread together with the cells. Okay, these are the typical profiles obtained with the simulations. Here you have concentration profiles, and here you have the current in black, electrical current, and these are the catabolic rates. Red is oxygen reduction, blue is sulfide oxidation. So practically what you see here is that you have a sulfide oxidation zone here, where you have this reaction, and this also corresponds to this bend in the sulfide profile. Sulfide is only diffusing here. That's why it's a straight profile, probably you know this. It's just diffusion. But here where it bends, you have consumption. And there is nothing here left.
What if you remove the insulator effect? What, have you done that?
To rebuild the insulator?
No, remove the insulator.
Oh, no. I'm not sure if these are insulated or not. Mm-hmm. There may be a loss of electrons on the way. So in this case, electrons are produced here, and you see from the current increase. Electrons in this part from sulfide oxidation are given to the wire, and the current increases. Then nothing happens here. The electrons are just transported. Ohm's law, constant current, and then electron flow drops because you have oxygen reduction here. If you remove the insulator, you would have electron loss all the way. So probably, I don't know, some other.
What do cells live off in the middle?
Yeah, that's the best question I think you could ask here. We don't know. How do they survive there? They just have some, I don't know.
They don't have to grow and divide.
No,
but they
have no energy.
They may die inside the sheath, but still the conductive structure remains. For how long does it remain like this? It should decay in the end if it's not maintained by the cells. No, it's cytochromes or so. they would be degraded probably. So what happens in the middle, we don't really know. We try to put decay as well. Okay, so this is the oxygen reduction zone, the red one, and you have here acidity production. You don't see very well this gray profile. This is the pH calculated, more acidic. On top is more alkaline. Electron consumption here, and this is just conduction, what I told you. And yeah, this would be kind of, development of these profiles in time. The green part here is the filament, which grows. So if I can play this again, you see how, okay. So this is the initial situation, trivial sulfide oxidizers. They just grow where oxygen and sulfide meet. And then you see here, this part, now the two zones separate. Sulfide is here, quickly consumed, electrons transferred here. At the end, there is conduction limitation. You see that even if the filament has grown like this, the reaction takes place still here. So you see this much better if I show you also the other one. So this is sulfide oxidation, this is oxygen reduction. As the filament grows, the electrochemical zones become wider. And you can imagine that the longer the filament, the more resistivity it has. So there is kind of an optimum here where they can stay, and then nothing happens. So I think we can conclude that we would have three regimes here. First, we have the biomass limitation, of course. The filament is not long enough. Then we have diffusional limitation. You see how oxygen penetrates here, and practically this is the active part. Only the cells here perform oxygen reduction, and the cells here perform the sulfide oxidation. What happens in the middle, I don't know. That's one of the unanswered questions. I would like to work with the Danish people, but never had the chance to present to them. Maybe we send this. Or maybe they will review our paper. It was sent last week. And then you end up with conduction limitation. If the filament is too long, the resistance is too much. And actually, there seems to be an optimal filament length and also an optimal conductivity for this. So if the filament is not conductive, then you don't have this separation of zones, just normal SOBs. If the filament is too conductive, there is no gain anymore, because you really need to have this difference in potential developing between the two ends of the filament. This is done together with Cristian Picioreanu, a PhD student from Brisbane, and this is his supervisor, probably you know from the anaerobic digestion work, Damien Batstone. There is one more thing. So we can somehow explain the concentration profiles, but the growth rates that we calculate are lower, about 3 or 4 times lower than observed experimentally, which means that the cells either have a higher yield for whatever reason, or they supplement their growth with other electron donor. So it's not only the sulfide, but maybe they can take iron or whatever. And it also means that there may be organic carbon involved here. It's not only autotrophic. Maybe they can grow in a heterotrophic way.
May I ask a question?
Yeah.
If they would use inorganic carbon, then their inorganic carbon uptake would also affect pH, right?
Yes. 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 If you have enough inorganic carbon, it won't affect if it is buffered enough. But if it is only a small amount of bicarbonate, let's say, of course it will be consumed, and then you have larger pH drops. Yeah.
Okay, but in this case, the pH changes are mostly driven by the electron, by the
yes,
the other oxidation.
by the other, yeah. And also, if you have organic carbon, also the oxygen consumption rates should change. Oxygen will oxidize this organic carbon. So it's still work to be done, of course. Anyway, this is the conclusion here. So I think the microbial fuel cell hype is somehow over the top now, and people are trying to find other systems. But I think still the intercellular electron transfer mechanism, or mechanisms, this remains very interesting scientifically. But I'm just an engineer, you know.
But that tells you that it is possible to have sort of a very close separation.
Yes. Somehow it's possible. If you can build some biofilm with these nanowires, it should be possible. Yes. Well, that's the end, of course. I wanted to put here two people who contributed to this. One is Madame Curie, who funded my stage in Newcastle, and this is, of course, the guy from Newcastle who actually recommended me for the Marie Curie. It was quite a productive collaboration with their group. Well, that's it. Sorry, this was a bit too long, but it's still fun.